that in whatever way is most useful for you. Okay, so we'll be continuing now today with more questions on what does the ectoderm give rise to in the body. And the last time we started talking about the central nervous system, and we're going to continue that theme today and then get into eye development. So one thing about the central nervous system, if we can say anything about it in the adult form, is that it certainly is complicated. There are hundreds of different types of neurons, and their arrangement within the brain and the spinal cord is, magnificently complex. And so for the developmental biologist, this sets up sort of an interesting struggle that we have to try to wrestle with to figure out is how do we set up such a system as this, where we could get so many different types of neurons or glial cells, how they come to make the connections that they do. And some of these cells, like the Purkinje cells, they make a spectacular number of connections. What about all these glial cells? So the brain makes 10 times as many glial cells as it does neurons, and what are they actually doing? And then what kind of genes are controlling this whole process from the embryo to the juvenile to the adult stage to control the formation and the integration of these neurons in glia? So there are a number of different questions. And in fact, the field of neurobiology in general is, I think, at a really hot time right now. So we have a lot of anatomy that we've known for some years. Now we have the genetics coming on with more genomic sequencing projects. And we have imaging coming on with better tools to try to label and track these cells during development. So I think you're going to see within the next 5 or 10 years, the continued trend of a real growth in the field of neuroscience, and this includes developmental neuroscience. So the system is complex, and yet, in a way, the parts list is fairly simple. So what we have to grapple with is neurons of various different types, and their support cells, which are glial cells. So these come in some different categories. Neurons can either be excitatory, that is stimulating the excitement of muscles or other neurons, or inhibitory. So some neurons tend to dampen down the process. And this kind of reflects that ongoing scheme that as you think about building a system during development, sometimes you take proactive types of gene actions, and sometimes the gene actions are more inhibitory in nature. And biology uses both of these to play off of each other and get a finer level of control. Then we have a few different types of cells that function as glial cells, and we think these mainly have some sort of protective and homeostatic type of functions within the organism. So oligodendrocytes, for example, these are involved in myelination, which involves the oligodendrocyte wrapping around a neuron. And so let's see, in our picture here, the purple cell is the oligodendrocyte. You can see how it's extending some processes that wrap around the axon of this blue neuron here. And that could serve to insulate that axon and protect it from maybe either damage or any kind of crosstalk between electrical signals that you would not want to happen. Okay. Oligodendrocytes can also be important for neuron survival or for cell replacement if some cells have to go in and replace cells that have died. Then astrocytes are a particular class of glial, so they have their own job to do during development, and they're especially important in the blood-brain barrier and in buffering. And then there are a whole group of cells called microglia that we actually don't really know exactly what they do in the organism. So they're not really glia, and there's some suggestion that they may have roles that play in immunity, but on the whole, there's still a lot to learn about this. So there are groups of cells here with different cell fates and different jobs to do in the construction of the brain and the spinal cord. And I'm going to kind of pick out a couple of paradigms relate to building the brain and the decision about neuron cell fate versus something else, and use this in the context of development. So the first one is this, cell fate decisions. So both in Drosophila and in vertebrates, we can, at early stages, point to precursor cells in the ectoderm. So this fits with our theme of ectoderm as a germ layer, what is it going to give rise to? We have some cells there that have maybe been patterned to take on more of a neural cell fate as opposed to an ectoderm cell fate. So in the course of living out this transition, they may go through a couple of different choices along the way. And one early choice is, are we going to become epidermis and some derivative of that, or will we become neuroectoderm? And remember when we talked about the frog, on the top of the archeneuron, that involuting tissue, there were signals going from mesoderm up to the overlying ectoderm at the root of the archeneuron, and that was predisposing that particular portion of the ectoderm to take on more of a neural cell fate, so that's what the neuroectoderm is up there. So then these cells that have received that early propensity, they are going to then make the choice to either go ahead and differentiate as a neuron, or to stay as something that looks a little bit more like a stem cell, and then could go on to make additional divisions and give rise to either glia or neural precursors. So there's a series of sort of bipartite choices, one thing or the other, as we go along the way, and then finally the cells adopt the final cell fate. And Drosophila looks very different in terms of the adult central nervous system, and yet when you think about how are they building their nervous system, in a sense you can see some parallels. And one of this is this series of bipartite choices, and it begins with the choice of hypoblast. So this is a cell fate that's mainly kind of epidermal in nature. The hypoblast cells are around the outside of the body, and I'm not completely sure, but I think they're involved in secreting cuticle, which is the hard insect integument that protects the outside of the insect. But for any rate, they're definitely near the periphery and basically epidermal in function in Drosophila. So the cell could either become hypoblast, or it could become neuroblast. And this phrase blast is often used in words that are associated with a precursor cell. So these are precursor cells that are going to give rise to neurons. And you see additional divisions where they either then give rise to a daughter that takes on that cell fate, or gives rise to just another neuroblast that would divide again and act more as a stem cell for this population. So there are certain parallels we can see between Drosophila and vertebrates, and this then gives rise to the possibility that we might see some similarities in mechanisms and possibly some similarities in genes using that the organism is using to get this job done. So where are the neuroblasts coming from in Drosophila? And this is a cross-section through the Drosophila larvae, and it's at the time of gastrulation. And so gastrulation, of course, is setting up these germ layers originally within the embryo. And one of the places that gastrulation occurs in Drosophila is right along the ventral side, along the anterior and posterior axis. You get this involution of tissue along the larvae, and that's shown here. And that involuted tissue will become the mesoderm. And then on the outside, we'll have the ectoderm, and especially here on the ventral side, these blue-colored cells are the Drosophila neuroectoderm. So these are the cells that are ectoderm in nature in terms of their germ layer, but they have a propensity to take on more of a neuron cell fate. And especially right down the midline, these dark blue cells, or in black on this picture here, these are especially going to become neuroblasts, and you can even see these eventually will delaminate from this epithelial sheet of cells here and some of these will migrate into the interior of the embryo. So that's just a little bit of a background for what we're going to talk about, and also to relate that to something we talked about earlier, these dorsal ventral polarity genes, the parallel genes, and the segment polarity genes. Those are all transcription factors, and they're going to be important in establishing this beta-2. So it's a hierarchical sort of thing here. We have certain territories set up in the embryo, and then those transcription factors activate their downstream targets, which are involved in more specific cell fates. So one of the specific cell fates here is neurons, and those upstream factors are helping cells make that decision. Okay, so just to tie it all together. Okay, so these cells in the neuroectoderm region, we can think of them as being pluripotent cells. So they're not, they have a certain limited number of choices now that they're going to make, and eventually take on these discrete fates. And we've touched on some of these concepts before, but I haven't talked about competence as much. So competence is going to refer to the ability of the cell to respond to signals that it might see in its environment, such as paracrine factors or juxtacrine factors or even endocrine factors that are coming from other cells and contributing to the decision of that cell about what its cell fate would be. So if it's not competent, it may not be able to hear these signals coming from other cells, or maybe not able to respond to them. So that's going to limit what kind of cell it would ultimately be able to become. So specification is also sort of overlapping and integrated with that idea, and the cells starting to take on their first information, and then sort of progressing into the differentiated phenotype, where the neuroblast might go ahead and make a neuron that has some function to it. So we're interested in mechanisms. So a lot of these little kind of stories that I give you are sort of very well known within the developmental biology community, and they're interesting in and of themselves. But the reason they're interesting is because they show us a way that this could happen. So it's almost like if you went to art school and they showed you a technique, make a bunch of different colored dots. Each dot is just one pure color, but when you put them all together, you get a bigger picture. So that's sort of a new idea, a new way of thinking about how you might construct something in art. And this kind of thing as well in development, just showing you a possible way that we could construct a cell fate or an organ or a tissue type. So here are some of the things that we learned from doing mainly genetic screens. So we put it out there, okay, organism, we're going to mutate you, give us some phenotypes, show us what genes are important for these processes, and let us figure out what these genes are biochemically and put them together into a story. So screens were done to try to find genes that would be needed to build the central nervous system, genes needed for the neuron cell fate or the glial cell fate. And not just can we get any neurons, but can we get the proper number of neurons within this organism? And can these cells partition appropriately into neuron versus glia cell fates? And so from these screens, we classified the mutants really into two categories. And the first one are genes that we would classify as neurogenic genes. And this is a set of genes that is really, their function is to govern how many cells take on the neural cell fate. So it's about like a gateway, getting the proper number of cells into this pathway to become neurons. We don't want too many, and we don't want too few. We just need exactly the right number. So this is governing that gateway, and there's two special genes, Notch and Delta. And this also kind of gives me the chance to introduce these genes because they're going to pop up, in other organ systems and other species, not just Drosophila, and are important and key players in several developmental processes. Both Notch and Delta are transmembrane proteins, So we'll talk about what they do in terms of their biochemistry. Then the second category of genes that came out of these screens are called the proneural genes. And these are involved in the ability of cells to have confidence to become neurons. So involved in their ability to respond to signaling from the neurogenic genes that says, okay, you are a lucky one, you have been chosen to become a neuron, go for it. And to go for it, you need these proneural genes. Biochemically, what they are transcription factors of a particular class called the basic helix loop helix. So they're just classified based on their structure. And of course, as transcription factors, they'll have a number of downstream targets that they regulate and complete the cell fate through the expression of these downstream genes. So in general, the proneural genes, they're going to be targets of Notch delta signaling. So the Notch delta part happens first, and then as a consequence of that, in the nucleus, that information is passed on down to activating some of these proneural genes. And then they, in turn, take it the next step further to implement the neuron cell fate. So in Drosophila, there's, as we have maybe mentioned before, this tradition of naming the gene based on the mutant phenotype. So recall when we were talking about dorsal, we named that gene based on a mutant that looked very dorsalized, that had an overrepresentation of dorsal cell fates. And the Notch gene originally got its name through this phenotype shown here. And it's a wing phenotype, where instead of the wild type wings developing, you would get these mutant wings that look like they had a notch or a bite taken out of them almost. And this is actually only a partial loss of function because it actually did survive to adulthood. And notch is going to be needed at several different times during the life of the fly. But this is how it got its name, this wing phenotype. So the wild type wing is very, very standard in Drosophila. It's always the same shape, the curve of the structure, even the veins, even the cross veins that are built in these wings are very standard from one individual to the next. So if you see any sort of variation on it, even a little lift out of the tip of the wing, that qualifies as being a mutant phenotype. So Notch, would have a role to play in wing phenotype, but even looking earlier than that, in that ventral region where the neuroectoderm is for Drosophila, Notch is involved in making that decision of do we go neuron or do we go hypoblast? And it's a bipotential fate. That means two choices, one thing or the other, and you're either going to become a hypoblast cell or a neuroblast cell. So initially we have this field of precursors known as the neurogenic or the neuron-making ectoderm cells, and they have to make a decision, one thing or the other, and we need the proper number. So we need about 1 out of 4 to become a neuron, and 3 out of 4 to become a hypoblast. So to get this ratio, we need genes to carry out that function. So genes are going to say, we're establishing a 3-to-1 ratio of hypoblast to neuron. So if you have genes involved in making this decision, you could mutate those genes, and you could alter the outcome of that decision. So that's what's happened here in the Notch mutant, is everything went fine in terms of setting up the precursor cells, generating the neuroectoderm, and so forth. But what went wrong was at this gatekeeping point where we were supposed to only get 1 neuron out of 4. This time, everybody became a neuron. So we get this huge excess of neurons within the Drosophila embryo, and simultaneously an underrepresentation of hypoblast cells. So from the point of view of a developmental biologist, this is a kind of an interesting mutant to get because it's going to teach us something about how this decision is made to aliquot the proper number of cells to a particular cell fate. Okay? So let's what notch is. And it is a transmembrane protein. And in this picture, it's shown in purple here. And so it's going to be present there at the surface of these cells, these precursor cells in the ectoderm. And it's going to function as the receptor for delta. So delta is another transmembrane protein on neighboring cells. And just by convention, we commonly call delta the ligand, or the sort of initiator protein in this cell signaling pathway, and the notch is the receptor. So at this point, all the cells in the field there, all the cells in the neuroectoderm field, are expressing both delta and notch on their cell surface. And I'm only showing you one protein at a time here, but initially everybody is expressing both delta and notch. And so what will happen when the signaling occurs, so when delta and notch meet, interact as signals to each other, in the receiving cell, a portion of this notch inner domain in the cytoplasm here is going to be cleaved off. And so this little purple zigzag portion of the protein, that will go to the nucleus and interact with some other transcriptive factors, and then we get differential gene transcription within that nucleus. So, if you're talking about cells interacting with each other, it has to start at the cell surface, signaling has to move through the cytoplasm, and then effect a change in the nucleus at the DNA level. Okay? So, this is actually juxtacrine signaling, because we need the cells to be in immediate proximity to each other. So the cells basically have to be touching. And in this case, we're looking at two transmembrane proteins, but juxtacrine signaling could be anything where any protein at the surface, even if it was, say, extracellular and tethered, or anything like that, this is just a type of signaling known as juxtacrine. Yeah?
Would it be possible for the cell to accidentally signal itself, like folded back?
Oh, would it be possible for the cell to signal itself if it was folded back? Yeah, I don't see why not. I don't know that that has happened to my knowledge in any specific instance, but in theory I would think that that would work functionally. Yeah?
So then is notch considered necessary for the hypoblast making, since they took it away and you didn't make any?
Notch would be necessary, because if you take it away, you're going to get a mutant phenotype that is not consistent with wild type function to create a neuroblast. So notch is necessary for neuroblasts. Okay. I like the idea of trying to think about what we're talking about now relating to previous concepts. So if anyone else has a question along those lines, that is an excellent question to ask. Okay, so we got these cells. Both of them are expressing delta, both of them are expressing notch. So how's this going to work out for making this decision? So here's one thing. So when the signaling event occurs, in the one who receives the signaling from delta on the neighbor, that receiver cell is ultimately going to downregulate its own delta transcription. So as a result of this receiving cell, who was originally expressing delta, is going to tend to downregulate the additional expression of delta, and over time might decrease its own delta receptor or delta ligand expression on this transmembrane surface, cell surface. So it becomes a situation where if some cells gain an advantage and start to express more delta, they will tend to downregulate delta in their neighbors. So eventually, a few cells become predominant as the winners. The guys who are expressing the most delta continue to be expressing delta, and then their neighbors express less of it. So just keep that idea in mind. So delta gets turned off, and then also the proneural genes are going to be turned off in this receiving cell. So some cells make a lot of delta, and they tend to keep their own delta on and their proneural on. Those genes become, or those cells become the neurons. The cells that are making a little bit less delta, they get the delta gene turned off and the proneural gene off, and those cells become hypoblast cells. So initially, we might have an entire clump of cells expressing both delta and notch, green in my figure here. And then after time, you look again, and at later stage, now you only see fewer, or one cell out of a clump, or one cell out of 4 on average, continues to express these proneural genes, and these then become the neuroblast cell fate, and eventually neurons and glia arise. So there's an element of time here where cells are sorting things out. So, oh, question.
I'm sorry, just that last mechanism you talked about, that's what sets up that 1-to-3 ratio?
That's what's going to set up the 1-to-3 ratio. And so this brings us to a concept here, a new concept called lateral inhibition. And this is how it works. Initially, we have a field of cells. The cells in that field are called the equivalence group because initially they are all equivalent in their ability to establish this particular cell fate. So initially, in theory, any one of these cells could become a neuron, and which ones actually do are going to be the result of these lateral inhibition events that happen between the cells. So this starts out, begins to change when one cell or some of the cells, a few of the cells, begin to express more delta. And we don't really know exactly how this happens. It could be stochastic. So stochastic is a term that we use in biology to call events that are sort of random. Like it's not totally random, but the system is sort of set up that generally a few cells just happen to make more delta, and you can't exactly pinpoint by genetics exactly which cells those will be, but in general a certain number of them do. So those are going to be the dark red cells. And so those cells making the most delta, they're going to tend to downregulate delta in their neighbors. So over time, the neighbors of the dark red cells are going to be pink, and then eventually they'll be white because they start to make less and less delta. So this, eventually the outcome of this will be that on average, 1 cell out of 4 will be a delta continued expression cell, the dark red cell, and those cells will become the neurons. Also, these neuroblasts, or these dark red cells, are going to be spaced appropriately, and that's because of this lateral inhibition due to juxtacrine signaling. So one delta predominant cell, it can really only affect its immediate neighbors. So it's not like it's going to dominate for 10 cell widths across the field. No, it can only downregulate delta in those immediate neighbors. So this works out well for establishing just a select group of cells as the ones who make delta and who will become the neurons. And this sort of mechanism of lateral inhibition, this can be used in various different situations by vertebrates or invertebrates. And one place that we see it would be in the pancreas, where you have to make a decision to become an exocrine cell or an endocrine cell, just two different types of cell fates. And this happens in a similar sort of way, where initially we have an equivalence group, and then eventually they get sorted out that only selected cells become one of the cell fates. Okay, any questions on this? All right, great. So here's a picture. I believe this is in immunohistochemistry, and we're looking at expression of a proneural gene. And initially you can see broad clumps of these neuroectoderm cells, or everybody is expressing the proneural gene, just like everyone is expressing the notch and delta. But then after time, this is shrunk down to now individual cells are continuing to express proneural genes, and all their immediate neighbors are not expressing the proneural genes. Okay, so I just like to show you a little bit of evidence to think about how we know what we're trying to talk about here. And this is one way. We look at markers for these different genes that were identified on the screen and see when and how these genes are expressed. Okay, let's think about the mutant phenotypes now. So we have these two different classes, and their neurogenic genes are notch and delta. The proneural genes are these beta HLH transcription factors. There's some of the names. Don't ask me where they came from. I don't know what those things mean, Scoot. But the mutant phenotypes are opposite here. So in the case of the neurogenic genes, if you mutate these, you're now going to get too many neurons because they're involved in the gatekeeper function of deciding how many neurons should enter the pathway. And if you break that, you could potentially get, say, too few neurons altogether, none of them in the pathway, or the opposite situation where now everybody went into the pathway. And then the mutant phenotype for the proneural genes is too few neurons. And so based on that, we can think about then what went wrong. What does that teach us about the wild type function? So in the proneural genes, these are needed to initiate or promote the process of neural cell fate over hypodermis. And these guys are more in the realm of competence, of being able to respond to that signal and carry it through. So this morning I was thinking about, if I could think of an analogy that might help you guys understand or grapple with or remember this. And I kind of came up with the idea of saying, let's train some students to become veterinarians. And so the veterinarian would be your differentiated cell fate akin to a neuron. And we have a process initially to sort through the applications. So of all these 200 students that want to enter the program and become successful veterinarians, we need to select those students who have the best potential to be successful at this. So if that process broke down, if we had a very lackadaisical admissions committee, perhaps we would now get 400 students entering the program, and that would be detrimental to the eventual success of all of those students or to the school as a training mechanism. So we need to have some way to regulate the proper number entering the program. Then we also need to have a mechanism to make sure that those students that get in the program get adequate